Okay, let's travel further down the collecting duct. We will come across what are called intercalated cells. These cells come in two types, type A and type B. And these are involved in acid-base balance. The fact that they're type A and type B is actually convenient. Type A, as we'll see, will be activated by acidic conditions, and type B will respond to basic conditions. So let's look and see how this will work. Maybe let's look first at a type A cell. First of all, we need to recognize that acid-base balance is an active process and therefore requires energy. There are two key drivers that facilitate this process, and they're both ATP-driven pumps. In the type A cells, these pumps are going to be on the luminal surface of the cell. So here's one of them, driven by ATP. I have another one down here, also driven by ATP. This top one is simply a proton pump, and it will pump H plus out into the lumen to be secreted. Now that makes sense because we said earlier type A is activated by acid, and so the job of this cell is to pump acid out into the filtrate where it's going to be excreted into the toilet. The other one does something very similar. However, while it's pumping H plus, it does that while simultaneously exchanging for potassium ion. This is interesting, but it makes sense from an electrogenic point of view. The upper pump is electrogenic. If I run that a lot, that's going to create a hyperpolarized effect on the cell as I remove these H plus ions from the inside of the cell. However, the second pump is not electrogenic. For every positive ion that leaves, I'm bringing a positive ion into the cell. Now, how might this actually work to reduce acid in the blood? Remember from previous work that the principal component of acid in the blood is carbon dioxide because of its relationship with H plus and bicarb ion. So as carbon dioxide readily crosses membranes, carbon dioxide will freely come inside the cell. Especially if it's high, that develops a concentration gradient. Once in here, carbon dioxide will bind with water to generate H plus HCO3 minus bicarb ion. This is just standard bicarb buffer equation. If CO2 is high, then that's going to drive this reaction, and we're going to have increased levels of H plus and HCO3. The H plus, of course, is just going to be pumped out here and here. Now, very similar to what we saw in the red blood cell, this band 3 transporter right here on the basolateral surface can be used as an antiport exchanger. I'm going to use the gradient of HCO3 to actually pump chloride into the cell, again, specifically for electrogenic purposes. Lastly, it's important to recognize that, similar to what we saw up here in the principal cell, we have potassium leak channels. So, as this potassium is pumped into the cell, it's going to leave the cell down a concentration gradient. This is interesting because we know that in acidic conditions, if you're diagnosed with acidosis, that potentially could lead to hyperkalemia, because in acidic conditions, these pumps, specifically this bottom ATP pump here, is removing a lot of the potassium that was secreted or excreted into the filter, into the urine, is being recovered now by this pump, and going down its concentration gradient is brought back into the blood. So acidosis can lead to hyperkalemia. In other words, if I decrease the pH, that could lead to increased K+. Okay, let's look at the other type of cell really quick. The type B cell, we'll draw it right beneath. And what you'll see is that it's nearly identical to type A in every way, except it's backwards. In other words, those ATP pumps, now instead of the luminal surface, are on the basolateral surface. But they work the same way. We have H+, that can be unilaterally pumped across, or down here, H+ that is exchanged for potassium coming into the cell. It's the same type of process where we go back to CO2, which is freely diffusing into the cell. We know that that is going to separate into HCO3 minus plus H plus. The same kind of scenario exists where I can take that bicarb and run it in an exchanger for chloride. Recognize what we're doing. In this cell, we're getting rid of the bicarb because the bicarb is the buffer that creates a basic environment. So as we pump bicarb into the filtrate, we're recovering, up here we're recovering H+. It is important to recognize that we also have this leak channel for potassium, but now it's on the luminal surface. So in basic conditions, I need to decrease bicarb and increase H+, and we can see how that happens. Importantly, in these type B cells, very similar to what we described above, but just opposite. In alkalotic conditions, in other words, if the pH is high, that could potentially lead to a hypokalemic state. Simply because this pump right here is running a lot and pumping a lot of potassium out into the filter, not at the filtrate, but the excrement. Because the activation of this ATP pump is driving the movement of potassium from the blood to be excreted by the kidney. But we also see a little bit of a twist. This wasn't true for the type A cell, but we see it for the type B cell, a scenario where decreased potassium, in other words, a state of hypokalemia, could actually induce a state of alkalosis. Because, going back to that same pump, if I don't have K+, if that has been reduced, then that ATP pump won't run. It needs the K+ in order to do its job. Without the K+, the ATP won't run, which means I can't pump the H+ ions here back into the blood. And without that, I can't really drive the whole buffer system that's going to bring in the bicarb and allow me to pump that out. And so that's something that physicians need to be very careful with as they regulate acid-base disorders and look into concentrations of potassium.